Let's quickly look at what we will cover in this video. So first we'll look at how muscles work together in antagonistic pairs, using the biceps as an example. Next we'll take a look at the structure of muscles. This includes key structures involved in muscle contraction, such as the sarcoplasmic reticulum. Then we'll take a deeper dive into the ultrastructure of myofibrils, as shown by this image here. And then third we'll take a look at the neuromuscular junction, and specifically how this relates to muscle contraction. Note there is a whole separate topic on synapses which covers this in more detail, so feel free to skip this if needed. And then finally we'll take a look at the sliding filament theory, which is a deeper look at how actin and myosin and other key players, such as ATP and regulatory proteins, interact to bring about muscle contraction. The first concept to understand is that muscles work in antagonistic pairs. Let's look at a bicep curl to see how this works. Now I'm using a very basic illustration of what's meant to represent an arm. Here the group of antagonistic muscle pairs includes the biceps and the triceps. Another key structure to be aware of is the tendons, which connect and transmit forces from the muscles when they contract to the bones, allowing for movement. You may have heard of sports injuries such as Achilles tendonitis, which is inflammation of the tendon. And make sure you don't confuse tendons with ligaments. Ligaments connect bone to bone, and these ensure the stability of joints. Similarly, you may have heard of ligament injuries in sports such as ACL or anterior cruciate ligament. Now let's see how the biceps and the triceps work together as antagonistic pairs. When the bicep contracts, this is known as the agonist here. The tricep does the opposite and relaxes, which is known as the antagonist. And you can see that the lower arm is pulled upwards. And the opposite way round is when the tricep contracts, the bicep relaxes. And you can see this time the lower arm is pulled downwards. And in real life, the bicep contracts, the tricep would be relaxed. And then the tricep would be contracted and the bicep would be relaxed. Now let's continue with the bicep and try to understand what's happening at a structural level which is causing the muscles to contract. Skeletal muscle is a highly organised tissue. I'm going to use a set of cylinders here just to try to show at a high level how this muscle is organised. Firstly, muscles are made of bundles called fascicles, which are encased and separated by layers of connective tissue. Then these fascicles are made of muscle fibres, and again muscle fibres are separated from each other by connective tissue. And finally, these muscle fibres are made of myofibrils. A single muscle can contain hundreds to thousands of these myofibrils. Interestingly, if you are more trained, i.e. you've done more bicep curls at the gym, you can have more myofibrils than someone who hasn't hit the gym in a while. Now I'm going to look at the structures of the muscle fibre in more detail using this illustration. The muscle fibre contains lots of nuclei, as you can see by the purple circles on this image. Why would muscle fibres need lots of nuclei? Well, muscle fibres need to produce many proteins for contraction in the form of actin and myosin. The second thing to call out is that the muscle cell has lots of mitochondria. Mitochondria are required for energy production, which is especially important in muscle cells. We look at the role of ATP in the muscle contraction later. As I mentioned previously, muscle fibres are made up of myofibrils. There can be hundreds to thousands of these. And myofibrils are made up of sarcomeres, which are repeating units and the basis of muscle contraction. I.e., when the sarcomeres contract, this causes the entire muscle to contract. We'll come back to the structure of these and how actin and myosin overlap later. Next is the sarcolemma, which is the membrane of the muscle fibres. It's important to note that the sarcolemma does not surround each of the myofibrils, but just the overall muscle fibre. Next are T-tubules, which are deep projections of the sarcolemma, almost like tunnels. You can see on the image with the yellow circles. They actually surround the myofibrils and are really important for transmitting action potentials from the sarcolemma, that membrane, to the sarcoplasmic reticulum. And finally is the sarcoplasmic reticulum, which is shown by this green mesh. This is responsible for controlling the calcium release, and once an action potential reaches from the T-tubules, it releases the calcium, which then causes muscle contraction. Now let's look at the myofibril in more detail, specifically the sarcomere. Our understanding of muscles came by observing these myofibrils under a microscope. Scientists observed two main protein filaments, the thin filaments of actin, you can see by the blue lines, and the thick filaments of myosin, you can see by the thicker purple lines. This image shows you an example of one sarcomere unit where you can see the actin overlapping the myosin, but in reality there would be many repeating ones. Now I'm going to introduce some lines and bands you need to be aware of. These are quite difficult to remember and originally derived from Greek and German scientific terms. I'm not going to try and pronounce them here, but feel free to look them up. So first are the M lines, which denote M for the middle of the sarcomere and attach to the myosin. Next are the Z lines, which are the boundaries of the sarcomere. Then are the I bands, which are made of just actin, and you can see by the blue filament here. As there is only one filament here, just actin, these will appear lighter if you viewed them on a microscope. Next is the A band, which is both actin and myosin overlapping. These will appear darker on a microscope as there are two filaments, you can see in the image, actin indicated by the blue one, myosin indicated by the purple one. And finally there is the H zone, which is just myosin. Again, as there's only one filament, this will appear lighter on a microscope. Now we're going to look to see what happens to these bands when the muscle contracts. Have a look at this image to see which bands are changing in size. The first thing you notice is that the actin and the myosin filaments are sliding over each other when the muscle contracts. Please note actin and myosin are not shortening, only the sarcomere has shortened. The first thing you might notice is that the Z lines have moved closer together. This shows that the sarcomere has shortened. Next, the I bands, which are made of actin, have also shortened. You will see the width of them has definitely narrowed from the previous image. Next, the H zone, which is made of myosin, has also shortened. But the A band remains unchanged. If you look at the size of the arrow under the label A, you'll see it's the same size as the image before. What triggers muscles to contract? We're going to take a quick look at the neuromuscular junction to show how the release of calcium controls this. To note, there is a separate topic which goes into more detail on synapses. If you already know this, feel free to skip ahead to the sliding filament mechanism. First, let's start by labelling the key structures involved in the neuromuscular junction. First, we have the presynaptic cell, which here is the motor neuron. Then we have the postsynaptic cell, which here is the muscle cell. If you think about the word neuromuscular junction, it's the specialized synapse between the motor neuron and the muscle cell. Next is the sarcolemma. Remember, this is the membrane of the muscle cell. Then we have the acetylcholine receptors, which are on the sarcolemma. And then finally the synaptic cleft, which is the gap between the pre and the postsynaptic cell. Now let's go through the steps of what happens at this neuromuscular junction and how muscle contraction is brought about. First, let's imagine you want your bicep to contract. I'm simplifying this, but essentially a signal is sent from your brain via neurons until it reaches the neuromuscular junction. The signal arrives first as an action potential at the axon terminal or the end of the motor neuron. This causes the opening of the voltage-gated calcium ion channels, which release calcium into the presynaptic cell. This causes the acetylcholine vesicles to then bind to the presynaptic membrane. This releases acetylcholine into the synaptic cleft via exocytosis. Acetylcholine then diffuses across the synaptic cleft and binds to the acetylcholine receptors on the sarcolemma. This causes a sodium ion influx and depolarization of the sarcolemma. This depolarization, if the threshold is reached, causes an action potential which travels down through the T-tubules, these are the deep projections of the sarcolemma, and this causes the sarcoplasmic reticulum to release calcium. This calcium causes muscle contraction, and you should be able to see the sarcomere shortening. Next, we're going to look at more detail in terms of how the sarcomere contracts, specifically the sliding filament mechanism. Now let's introduce the main players involved in the sliding filament theory. So first we have actin, which is a thin filament. This is surrounded by the regulatory protein, tropomyosin. This means it regulates or controls muscle contraction, and we'll see how this works in a moment. We also have troponin, which is another regulatory protein. Although for the AQA specification, you don't specifically need to know about troponin, I think it's important to include for understanding the overall concept. We also have myosin filaments, which is, if you notice, a thicker filament at the bottom and then the myosin head sticking out of it. To note that the myosin heads are in this high energy and cocked position, and what all this means essentially is that they're ready to bind to actin and initiate muscle contraction. This is because they have this stored potential energy from earlier hydrolysis of ATP, and you can see ADP and phosphate attached to the head. We'll look at ATP hydrolysis because it's a step later. In this position, when the muscle is resting, the tropomyosin has blocked the actin-myosin binding sites. This means myosin can't bind to actin and muscle contraction can't happen. But when calcium is released from the sarcoplasmic reticulum, it binds to troponin. This causes the tropomyosin to change positions, exposing the actin myosin binding sites. The myosin heads can now bind to the actin, and a cross bridge between the actin and the myosin is formed. Next, the myosin head performs a power stroke, which pulls the actin filaments towards the centre of the sarcomere, releasing ADP and phosphate. You can see that the sarcomere has shortened. Next, ATP binds to the myosin head and causes it to detach from the actin. Rigor mortis is a condition where there isn't any ATP, so the myosin head remains attached to the actin, locking the muscle fibers in this contracted position. ATP hydrolysis can then occur, which releases energy to recock the myosin head back to the original position so that the cycle can continue. The cycle can continue, assuming that the muscle fibre is continuously stimulated by nerve impulses, as this triggers the ongoing release of calcium ions from the sarcoplasmic reticulum. Remember back to the neuromuscular junction. But the cycle stops when the muscle fibre is not stimulated anymore, and the calcium ions return back to the sarcoplasmic reticulum via active transport, which needs ATP. To wrap this up, I just want to summarise the role of ATP in muscle contraction, as this is something that often comes up. So the hydrolysis of ATP provides energy for the power stroke of the myosin heads, which pull the actin filaments towards the centre of the sarcomere. ATP is also needed for detaching the myosin heads from the actin filaments and resetting the myosin head to its original position. Finally, it's responsible for the active transport of calcium ions to return back to the sarcoplasmic reticulum. So this is the end to the video on muscle contraction. I thought I would leave you with this rowing boat, as it's often an analogy used to describe the sliding filament theory. See if you can figure out what the boat oars and the water is meant to represent.